Okay, so I'm gonna start. I hope y'all can hear me. But so I basically did a poster on how enzymes digest food. So I'm gonna read and then I'm gonna tell you a little bit about it. And y'all can ask questions if you're confused or you want to know more. So for the introduction, enzymes are one of the most important molecules. This is because they allow crucial processes like digestion, which we will be talking about, DNA replication, and energy production occurs quickly. Without enzymes, humans wouldn't be able to digest food, move correctly, make brain chemicals, or other vital tasks during the day. In addition, the symptoms like constant gas and bloating and stool changes, which we will get into more later, is a sign of a need in enzymes in the body. And a lot of times when we think of digestion, we just think of the stomach, but it involves much more than that. Um, Digestive enzymes are used for the digestion system to break down food and supply our body with the nutrients it needs. Enzymes use different processes to turn monomers into polymers. A monomer is like, think of a single Lego piece, it's just one thing, and then the polymer is like the finished product, so it's many monomers put together. They act as catalysts, which speeds up chemical reactions, and it does so at body temperature. The process starts at the mouth all the way through the large intestine. There are also different enzymes like amylase, protease, and lipase that help in this crucial process of human survival. So, before it even starts, you have to have enzymes. So, we're going to talk about how they start and how they form. They're made through specialized cells, which is a certain cell that does one specific job in the salivary gland, stomach, pancreas, and small intestine. The pancreas, however, is the powerhouse of digestion because it produces many of the important enzymes to break down what is eaten. In addition, there are different phases needed to make these digestive enzymes. This includes, sorry if I say this wrong, the cephalic phase, which is the sight, smell, or thought of food. There's the gastric phase, which happens when food arrives to the stomach. And then there's the intestinal phase, when the digested food moves through the stomach to the small intestine. Hormones like CCK provoke the intestine to release a large wave of powerful enzymes to finish digesting the rest of the food after it moves from the stomach. So more of how it works and what these different enzymes are. The amylase is made in the mouth and pancreas, so that's like the first step you could call it when you first start eating, and it breaks down carbohydrates. Lipase is made in the pancreas and breaks down fats. Protease is made in the pancreas and breaks down proteins. And the lactase breaks down lactose, and the sucrase breaks down sucrose, and that's basically just like sugars. And then as food travels through the body and many stopping points, it's important to know what's happening in your body if something isn't working right or you have symptoms that are abnormal. And that's what we will be talking about next, which is health concerns. Um, Disruptions can lead to malnutrition and or gastrointestinal irritation. Some symptoms can include belly pain and cramps, bloating, diarrhea, gas, unexplained weight loss, um, and unusual stools. If you don't get it checked out, it can lead to more serious conditions. Some digestive enzymes insufficiency can lead to lactose intolerance, which means one can digest lactase. One can't digest lactase. It's difficult to digest sugar found in milk and dairy products. EPI happens when the pancreas doesn't produce. Well, let me back up. EPI is exocrine pancreatic insufficiency, and that happens when the pancreas doesn't produce enough enzymes to the needed to digest carbohydrates, proteins, and fats. There are many ways to treat this, like vitamins, medicines, supplements, and different diets, depending on your situation and how serious it might be. It's important to track what you eat and take care of yourself before it gets worse. Moving on to the pictures, I put two on here. So the first one is like everything in the body that works to digest your food because there's many different parts. And then this picture shows how different sugars and molecules and stuff are made to digest. So this is how like sucrose is made. You have your bond and it goes to the substrate. It's like a lock and key type of thing because you can't, they all work for one different type. Okay, so it's like a lock and key. You can't have a different lock for a building and use it for your house. It's specific to that certain enzyme. So they come together at the active site. They bind and go through enzyme substrate complex, which is the second one, and they keep forming and going together. And then at the end, there's products are released, and that's what you get from the different processes made to make this specific molecule. I hope that made sense. I'm not sure. And then at the bottom, I have the references I use.
Yeah, you did an amazing job with this poster. It's really informative. And maybe a question to the audience or just everyone in this call. Before this presentation, did you kind of know what the pancreas is for? Because I feel like it's an organ that's so important, but not a lot of people know about it. So maybe you can react with a thumbs up if you knew what the pancreas was doing before. Let's continue with this poster.
Okay. This poster was made by me and Vaishnavi, and I'll start. The body requires a lot of energy. The digestive tract is how the body takes fuel from food. But how does it, or more specifically, its enzymes work? We have conducted a literature review on various sources to find how pancreatic cells make digestive enzymes and how these digestive enzymes work in the small intestine. Now, for the introduction, we did bullet points to save on space, so I'll start there. The digestive enzymes are proteins used by the digestive enzymes to break down food for, energy because the body can't use food on its own. And the stomach, small intestine, oral cavity, and the pancreas all make digestive enzymes. However, the pancreas is the one making the main producer of all these digestive enzymes, and it for its size is over 12 to 18 centimeters long. The pancreas is a dual function gland that supports digestion and blood glucose regulation. Its exocrine portion produces digestive enzymes that are released into the duodenum. And what the exocrine portion is basically the outer layer of the pancreas because it is what is secreting digestive enzymes into the duodenum, which is part of the small intestine. While the endocrine portion secretes hormones like insulin and glucagon into the bloodstream. Pancreatic digestive enzymes play a critical role in chemical digestion by breaking down large food molecules into smaller nutrients that can be absorbed by the intestine. Since these enzymes can digest proteins, they are produced and transported in carefully regulated forms to protect pancreatic tissue from self-digestion. Their production, secretion, and activation are tightly coordinated to ensure that digestion only occurs after the food reaches the small intestine. Exocrine portions are, as we know, exocrine portions are the ones producing digestive enzymes. These exocrine these exocrine oh sorry. These are called exocrine because the digestive juices that they produce flow into the small intestine rather than to the bloodstream. The exocrine portions of the pancreas are made out of specialized acinar cells. These are the cells that are specifically producing digestive enzymes for the body. These cells produce three main types of enzymes: lipase to break down fats, protease to break down proteins, and amylases to break down carbohydrates. These cells have highly specialized endoplasmic reticulum, which make it possible for the synthesis and the transportation of these digestive enzymes into the small intestine, where food can be digested. And the following portions are Vaishnavis. So,
Yeah, so moving on to the disorders of impaired enzyme production. Because this process is so tightly controlled, problems can occur if anything wrong happens with the process. So, one example is the exocrine pancreatic insufficiency or EPI. So, in this condition, the pancreas doesn't produce or release enough digestive enzymes, and as a result, food isn't broken down properly, leading to poor nutrient absorption. And patients can experience symptoms like weight loss, diarrhea, fatty stools, and vitamin deficiencies. Another example is pancreatitis, which is basically almost the opposite problem. So instead of the enzymes activating in the small intestine, they become activated too early while they're still inside the pancreas. And since these enzymes are designed to digest food, including protein, and the pancreas is made out of protein itself, they begin digesting the pancreatic tissue instead of the food, causing inflammation, severe abdominal pain, and impaired digestion. And these diseases highlight why it's so important that the pancreatic enzymes are produced, transported, and activated at exactly the right time and in the right location. So, we have differences in activation types. So it's critical for these digestive enzymes to be activated at the correct time and location. So successful digestion while protecting the body's individual tissues, as I just mentioned, pancreatitis. So this is why this is so important. So to attain this, many enzymes are synthesized in cymogens, which are inactive precursors. And these enzymes remain inactive until they meet at the site of action needed for. And a reference is stomach acid converting pepsinogen into pepsin to initiate digestion of protein in the stomach. And pancreatic enzymes are inactive until the small intestine is met with, where the enzymes are beginning to digest the protein, carbohydrates, and fats and other macromolecules. In comparison, some enzymes are energized through other regulatory mechanisms, and these can be binding to like helper molecules or responding to cellular signals. As enzymes are monitored for mobilization, the body can safeguard its own tissues from chemical breakdown while confirming sufficient digestion. And Shravya will be concluding this poster and describing the images as well.
Sounds good. So, to overall to explain these posters, here we have the structure of the pancreas. So we have the endocrine and the exocrine, where here each part is labeled. The pancreas is the inner part where it's yellow and it has a lot of green molecules. The this is there's also kind of a shape that looks like a stem. This is known as the bile duct, and it's from the gallbladder. We also have common bile dust, which is the end of the stem, and we have digestive enzymes located around the middle. We also have the duodenum of the small intestine, which is kind of the outer layer that's surfacing the digestive enzymes. We have the pancreatic duct, and we have the acinar cells secrete digestive enzymes, which are two, they kind of look like leaves on the stem inside the pancreas. So you can kind of refer to these illusions as labels of the pancreas. And we have pancreatic islets, and we have spleen, and we have pancreatic islet, which is alpha cells, beta cells, and exocrine alpha. And here we also have a figure, which is an electron micrograph of the pancreatic acinar cell. This micrograph is basically highlighting the critical cellular structures that we analyzed for synthesis, processing, and storage of digestive enzymes. For the left side, we had rough endoplasmic reticulum, and in the middle of this figure is the Golgi complex, and on the right are zymogen granules, and these are adapted from Gorelick F, Pondal SJ, Topazian M, and this is from the pancreatic physiology, pathophysiology, and the acute and chronic pancreatitis. The Gastrointestinal Teaching Project, American Gastroenterologic Association are all sources that and references that we used for this figure. And to kind of go a little bit in depth for these images, we also highlighted the difference in the activation types. So this is where enzymes um, are used as zymogens, and zymogens are basically inactive forms of enzymes, and they need to stay inactive until they reach the location where they're actually needed. So also a reference, an example that we had was the enzyme pepsin, and this is first produced as pepsinogen, and it's an inactive precursor. Once pepsinogen comes into contact with stomach acid, it's converted into active pepsine, and then it continues into breaking down proteins. And a similar process that we can note is also enzymes from the pancreas, which is similar to the figure that we had above. They are released as an inactive form and only become activated once they reach the small intestine, where they help digest the proteins, carbohydrates, and fats. However, not all enzymes rely on zymogens. They are activated sometimes through regulatory mechanisms, and an example is binding to helper molecules, and these are labeled as cofactors, or they can respond to specific cellular signals. And overall, these activation methods are they are there to control when and where enzymes work. And to finally conclude our overall poster, we have a kind of a quick um, note that you guys can take away from what we shared today. And the pancreas is a vital role in our digestive system, and this creates producing enzymes that break down into proteins that fats and nutrients can absorb. And something to note is that if the pancreas or enzymes are not functioning correctly, disorders such as exocrine pancreatic insufficiency and pancreatitis showcase the importance of proper enzyme production and regulation. And overall, this was our poster, and here are our references cited below our text. Thank you.
Thank you so much for presenting this poster. It was amazing, and I think we could all learn a lot.
Oh, okay. Okay, so in our body we have three main digesters, which are amylases, which begin their work right in the mouth. Uh, they're found in the saliva, and then they continue in the small intestine. So they digest the complex carbohydrates and starches. They are mostly found in bread and pasta, and then they break them down from starch to sugars, like glucose, for the immediate energy for cellular respiration. Then we have proteases, including pepsin, which will take the center stage in the acidic environment of the stomach and small intestine. Wait, and essentially what they do is they break down the tough and tightly folded chains of proteins from food like meat, eggs, beans into individual amino acids. And the body uses the amino acids to repair tissues and build muscle. Finally, we have the lipases, which operate within the small intestine to tackle dietary fats and oils, breaking them down to fatty acids and glycerol to support the cell structure, and they also provide long-term energy storage. And for those of you who don't know, enzymes are specialized proteins which speed up the rate of reactions in the body. And a fun fact about them is that they can never finish in the body. So enzymes work by digesting food because they are biological catalysts. So they act as molecular scissors, which speed up chemical reactions. So when we eat, the large molecules in our food cannot be directly absorbed by the body. So the enzymes solve this through a lock and key mechanism. Each enzyme has a unique shaped pocket called an active site, and it fits only one specific food molecule known as a substrate. So when the food molecule binds into this active site, the enzyme weakens the chemical bonds which holds the molecule together. So they split it apart into smaller pieces. And so because the enzyme, it's never going to be altered or consumed by a reaction, it's going to be immediately released. Um, and then they move on to digest the next piece that comes down.
Yeah, I think that covers a lot of the aspects of how enzymes work. I just want to add something about the lock and key model. That's something that appears over and over again in biology, so it's a concept worth remembering. But with enzymes, it isn't like that the key and the lock are like very fixed, but they also can change a little bit. So if the substrate binds to an enzyme, the the enzyme has to change the form just a little bit, and by doing that, it can like the the substrate binds, but then because it has to change the form a little bit, also certain bonds are weakened, which also can catalyze chemical reactions. Okay, thank you, Abiha, for presenting this poster. It's really great, and I think that this picture summarizes a lot of basically everything you have to understand about enzymes and how digestion works. Okay, you can continue writing questions in the chat. And if there are no questions, I'd like to ask the interns that have been working on those um, um, posters and also articles to to share some advice on conducting literature reviews. You can go ahead and answer the question.
Sorry, can you repeat the question?
So I think the journal interns can share a bit of their process of conducting literature reviews and the difficult parts of it.
Okay. I can't speak for everyone, but at least for me, the process starts with finding sources. Well, after you've had your research question, of course. For me, what I've been working on was the topic of regeneration. So often I've tried to find resources and like research papers online that speak about it. For example, I've had um, a couple sources that speak about how genetic engineering can affect regeneration in like certain processes, such as regenerating hair cells within like mice, and genetic, and it's also also its application in its use to regenerate failing bones. And from there, it kind of works like a synthesis essay for me, because you really only have to read the sources, and then as long as you understand what the sources are talking about, you can use it as evidence and analyze it to help answer your research question. And for the most difficult part, I'd say arguably it would have to be reading through the sources and finding out which ones truly answer your question.
Okay, thank you for sharing that. And maybe the other interns can share their thoughts too.
I honestly agree with Timothy. Like, it's hard to find the sources that are like real and not just spreading false information. So it's important to find sites that are real. Like, I don't use Wikipedia because anybody can change that. But like I used, for example, I use like John Hopkins medicine and that kind of thing. And I also like watching Amoeba Sisters for biology and that kind of stuff. But the hardest part, honestly, is just starting. Once you start, it's so easy. But like, it's hard to know what information you want to put, what you want to explain, like, because I feel like there was so much you could talk about, and we had to put all that information into a tiny poster, so it was kind of difficult. But yeah, that's my response.
Okay, thank you for sharing that. Has anyone else some thoughts about conducting literature reviews?
Yes, I also agree to what Timothy and Jazzy have shared earlier. I feel like it's really important to verify your sources, and something you can do is always check the time of publishing so you can see if this data has been reviewed. You can also you can also do comparison between multiple sources to check like is some older data similar to like what new scientists have studied recently. You can also check the authors to make sure that they have experience in what they've been working on or if they've been mentored by another scientist or another candidate. And I agree that starting is very hard because but once you begin the process, it's really easy to just continue and keep writing. And also make sure to have your own analysis done, like with all these sources. Don't just extract text from the source. Just try to make your own make your own like review and kind of share your own thoughts about what you've studied so far.
From my experiences, I feel like another difficulty is like to find real scientific papers that you can read because many are behind a paywall, which is very unfortunate. But there are also these preprint servers that contain the non um, peer-reviewed articles. I don't know if that's the best source, but that's at least for some sort of informal literature review, you can still get quite beneficial sources from. And tools like Google Scholar, but also um, there are um, like there are AI search engines that just cite scientific papers, and I think that's also a good use of AI and for such a project. But obviously, if you just get AI sources and don't go back to the real source, that is obviously not good.